Hello and welcome to Applied Anatomy and Physiology, AAP 301 for the College of Natural Health Sciences, Bermuda. My name is Dr. Delcina Bean Burrows, and this is lecture number 8. And in today's lecture, we will be discussing the neuromuscular and myofascial connections. The learning objectives for today's lecture are that at the end of today's lecture, students will be able to discuss the importance of understanding key neuromuscular and myofascial connections in the practice of manual therapy, describe the two neuronal loops utilized by muscle spindles to moderate muscle tension, explain how manual therapists can use their knowledge of reciprocal inhibition, stretch reflex, and gamma gain to reduce muscle tension, describe the different physiological mechanisms involved in the development of tender points and trigger points and what implications they have on manual therapy choices, explain the concept of tensegrity as it applies to the human body, explain the general location and function of the layers, bands, and planes of the fascial system, explain the functional importance of myofascial chains and how knowledge of these chains might affect therapeutic choices, explain the key mechanical properties of fascia and how knowledge of these properties might affect therapeutic choices, name the 4 types of mechanoreceptors found in fascia. Discuss how knowledge of the mechanoreceptors and smooth muscle cells in fascia might affect therapeutic choices. Describe the neuromuscular and neurofascial mechanisms related to maintaining posture, coordinating movement, and regulating muscle and motor tone. And finally, explain the difference between motor unit and muscle recruitment and between myofascial and kinetic chains. The key terms that we will discuss are alpha loop, deep fascia, fascial band, fascial plane, fascial tone, gamma gain, gamma loop, hypertonicity, interstitial myofascial tissue receptor, myofascial train, neurofascial loops, and neuromuscular reflex. We will also consider phasic muscle, piezoelectricity, plasticity, postural muscle, superficial fascia, tender point, tensegrity, viscotropy, trigger point, and viscoelasticity. So let's get started. Everything in the body is connected. As manual therapists, we manually manipulate skin, muscle, and fascia, stimulating the sensory receptors. The client's experience of relaxation is due to several factors. One is the resetting of the motor tone by accessing the neuromuscular loops. The second is by loosening, stretching, and broadening the connective tissues. And the third is by decreasing the sympathetic tone. So in terms of the neuromuscular reflexes, they control and coordinate skeletal muscle contractions. There is what is referred to as a simple reflex arc, and there is also a complex neuronal pathways which involve multiple interneurons in the spinal cord and the brain. This leads us to our discussion of reciprocal inhibition. Reciprocal inhibition is the reflex mechanisms that coordinates the effort between the agonist and the antagonist muscles. Muscles receive simultaneous signals. The agonist is stimulated to contract, and the contraction of the antagonist is inhibited. It can be utilized by therapists to relieve hypertonicity and muscle spasm. And here we see illustrated reciprocal inhibition for cramp relief. To relieve a cramp in the gastrocnemius, the therapist employs direct compression and reciprocal inhibition simultaneously. As the therapist firmly presses down on the cramp with one hand, the client is told to pull his foot against the therapist's other hand. This engages the antagonist muscles, which are the dorsiflexors, and inhibits the gastrocnemius. Let's now consider the stretch reflex and gamma gain. The muscle spindle has two distinct portions, the alpha loop and the gamma loop. It helps explain the development of tender points, which is T(e)ps, and muscle tension, as well as trigger points, which is T(r)ps, which is different. Now let's consider the neuromuscular neural loops. The muscle spindle uses two different neuronal loops to monitor muscle length. The alpha loop innervates the extrafusal fibers and manages the stretch reflex, while the gamma loop adjusts the tension in the intrafusal fibers to increase muscle spindle sensitivity to rapid lengthening. So let's consider the characteristics of tender points. Tender points may or may not be palpable. They are hypersensitive to mechanical pressure. There is no referred pattern of pain, and they are often silent until compressed. They cause the muscles to be resistant to stretch. Let's now, in comparison to the tender points, consider the characteristics of trigger points. Trigger points are usually palpable by way of a nodule in taut bands of muscles. They are hypersensitive to mechanical pressure. The pressure reproduces pain complaints in predictable patterns, and they usually cause the muscles to be hypersensitive to stretch. Let's now take a look at the concept of gamma gain. When the elbow is in the neutral position, the neurological report from both the biceps and triceps brachii muscles is low. When the elbow is flexed, the neurological report from the shorter biceps is decreased, while the report from the lengthened triceps is increased. During the shortened phase, the gamma loops of the spindles in the biceps increase their sensitivity to lengthening, which is called the gamma gain. Now let's consider gamma gain when the elbow is in a locked, resistant to stretch position. Due to gamma gain, sudden lengthening of the biceps, which is the weight suddenly dropped into the hand, it stimulates a stretch reflex contraction of the muscle even though it is not fully lengthened. The following table illustrates the characteristics of trigger points and tender points by comparison. The pathophysiology of trigger points is that the motor end plate dysfunction causes the calcium spill into the sarcomere, leading to actin-myosin bonding. In tender points, there is a high gamma gain in muscle spindle signal contractions in response to sudden lengthening. The characteristics of trigger points is that there is usually a palpable nodule in taut band of muscle. There is hypersensitivity to mechanical pressure. The pressure reproduces pain complaints in a predictable pattern. There is the cause of the muscles to be hypersensitive to stretch. But with tender points, there may or may not be palpability of a nodule. There is hypersensitivity to mechanical pressure. There is no referred pattern of pain, and it is often silent until compressed, and the causes of the muscle to be resistant to stretch is present. And here we see illustrated the tender point release. The location of a common tender point is the tensor fascia lata. To release the tender point, The muscle must be returned to a shortened position to desensitize and reset the muscle spindle. For this tender point, the primary position is hip flexion, while internal rotation and abduction of the hip are used as fine-tuning movements to find the best position of ease. The energy crisis related to trigger points is a self-sustaining cycle. The motor end plate dysfunction results in excessive calcium release from the sarcoplasmic reticulum, which allows actin and myosin bonding. The shortening of several sarcomeres in muscle fibers increases the demand for energy to sustain the contraction, while at the same time the energy supply is decreased due to the restriction of circulation from the contraction. In this trigger point release technique, the location of a common trigger point is the suboccipitals and their associated referred pain pattern. One method of desensitizing the trigger point is to curl the fingers up and under the occiput, placing moderate pressure on the points. Another method of releasing this trigger point involves placing a thumb over the point, then extending and rotating the head over the thumb to increase pressure and desensitize the point. Now let's discuss the inverse stretch reflex. The Golgi tendon organ, or the GTO, responds to increased tension by inhibiting contractions. Several forms of manual-therapy use stimulus to the GTOs to decrease muscle tension, such as the active release technique and the facilitated stretch technique. Now let's consider the fascial system. The fascial system is an individual connective tissue structure that can be considered collectively as a unified system. The fascial system includes all fibrous connective tissues. It is the unifying structural element for the entire body. It extends from the skin to deep within the muscles and internal organs. It makes the body strong, flexible, and resilient. Tensegrity is the dispersion of stresses throughout the system. And here we see illustrated the fascial layers. We see the meningeal layer over to the left most superiorly, and just beneath we see the lumbosacral aponeuronas. Over to the right most superiorly we see the pannicular layer, followed by the axial layer and the visceral layer. And as we just mentioned, the layers of the fascia consist of the pannicular or the superficial fascia, the axial fascia, the meningeal fascia, and the visceral fascia. Let's discuss the fascial patterns. There are superficial and deep layers of fascial system that are organized into the well-defined patterns that we see either vertically, in a spiral fashion, or horizontally. The patterns form multiple lines of tension. This helps the body to maintain its structural integrity, and it also helps the body to generate and transmit force for movement. Now let's discuss myofascial chains. Myofascial chains are the connective tissue links between muscles, bones, and fascial membranes. They are most vertically aligned. They form straight lines in coronal or sagittal planes, and they form spiral vertical lines that traverse all three planes. Treatment along the entire myofascial chain may be required. And here we see illustrated the myofascial chains for the lateral and spiral line. The lateral line includes the splenius capitis and the SCM, the external and internal intercostals, the lateral abdominal obliques, the gluteus maximus, the tensor fascia lata, the IT band and abductors, anterior ligament of the head of the fibula, and the peroneal muscles with investing fascia. The spiral line links the splenius capitis, the cervicis, and the opposite side of the rhomboids and the serratus anterior. And here we see from the anterior view the spinal line. And here we see illustrated the myofascial chains of the superficial and deep front and back arm lines. The superficial back line includes the tendons of the toe flexors, the plantar fascia, the Achilles tendon, the investing fascia of the gastrocnemius, the hamstrings, the lumbosacral aponeurosis, the erector spinae, and the scalp fascia. The superficial front line includes the toe extensors, the fascia of the anterior compartment, the patellar ligament, the quadriceps, the rectus abdominis, the sternocondral fascia, the SCM, and the scalp fascia. Now let's discuss the fascial patterns in terms of the horizontal bands and the planes. In terms of the fascial bands, 7 horizontal bands of superficial fascia are found around the torso. They add rigidity and contour to the body. 4 fascial planes are held within the cavities that provide structural support. Tension or restrictions can have profound effects on the structures supported. Here we see illustrated the horizontal bands. The body has 11 horizontal bands of superficial fascia that add contour and support to the torso. Now, in terms of the fascial planes, there are 4 deep fascial planes arranged horizontally across the dorsal and ventral cavities. So now let's take a look at the mechanical properties, the first being viscoelasticity. Viscoelasticity is the ability of tissues to extend and rebound rather than stretch and recoil. It is due to the organization and structure of the collagen fibers. Its increased exercise stimulates the fibroblasts to increase tropocollagen production. The frequency of manual therapy treatment sometimes is more important than the duration of each session. Now let's take a look at the mechanical property of thixotropy. Thixotropy is the ability of ground substance to vacillate between either a gel state, which is a viscous state, or a liquid state, the sol state. It occurs in response to either temperature or movement. Piezoelectricity is the next state we will consider. The connective tissue, which is subjected to mechanical pressure, produces small electrical charge. This current excites fibroblasts while exhibiting function of fibroclasts. Pressure and movement of manual therapy increases piezoelectricity of fascia, and it may contribute to the healing and softening and loosening of client sensations. Our illustration here shows the structure of a collagen fiber. Each collagen fiber is made of smaller bundles of fibrils. Fibrils are made up of bundles of microscopic filaments called tropocollagen. The spinal structures of tropocollagen is reflected throughout a collagen fiber, which is why collagen extends or unwinds rather than stretches when the tension is applied. And here we see illustrated the pin and stretch technique for the latissimus dorsi and teres major muscles. In illustration A, we have the starting position, while position B, we see the finish position. Let's discuss fascial plasticity. Fascial plasticity is the changeable, responsive, and adaptive nature of fascia. It is not fully explained by mechanical properties of fascia. Let's discuss the fascia as a sensory organ. Fascia is very rich in sensory receptors. There are 4 types of mechanoreceptors: the Golgi receptors, the Pacinian receptors, the Ruffini organs, and the interstitial myofascial receptors. Manual therapy is a primary treatment for chronic pain syndromes. And here we have a very nice table of the mechanoreceptors in the fascia, divided according to the receptor type, the preferred location, and the response to and the known results of stimulation, according to whether it is a Golgi organ, a Pacinian, a Ruffini, or interstitial type 3 and 4. And here we see the fiber distribution in a typical muscle nerve. A typical skeletal muscle nerve contains only a small number of somatic motor neurons and a larger proportion of autonomic, motor, and sensory fibers. Of the sensory fibers, appropriately 80% are interstitial myofascial receptors. In consideration of our pathology alert, fibromyalgia is a debilitating chronic pain syndrome. It is characterized by widespread musculoskeletal pain, disturbed sleep, persistent fatigue, and tender points. Pain receptors are in the muscles, joints, and fascia. Stress and emotional changes are often correlated to the intensity of fibromyalgic pain. Now let's consider the intrafascial smooth muscle cells. Smooth muscle cells are contained within fascia and are controlled by the autonomic nervous system hypothesized to produce fascial tone. They are the links between fascial sensory receptors, the autonomic nervous system, and the intrafascial smooth muscle. They help to explain the physiological effects of manual therapies. It also affects likely mediated through neurofascial loops. Also, there is decreased fascial tone, which helps to explain the increased flexibility and range of motion gained through manual therapies. The perimysium of skeletal muscle contains a high density of intrafascial smooth muscle cells and, like all smooth muscle, the autonomic branch of the nervous system controls these cells. The nervous system controls structural tensegrity for posture, balance, and movement. It is constantly shifting motor unit recruitment for postural muscles. The central nervous system is responsible for muscle recruitment to control the agonist and the antagonist for movement. The role of the brain differs according to the area of movement. And here we see illustrated tissue manipulation and palpable tissue response. Stimulation provided to the mechanoreceptors by tissue manipulation simulates both somatic and autonomic divisions of the peripheral nervous system. This causes palpable changes in the tissue that manual therapists often refer to as tissue release. Now let's discuss proprioception. Proprioception is the sense of body position, while kinesthesia is the sense of movement. Proprioceptors in fascia have a large role. Within the nervous system, a continuous feedback loop of sensation and adaptation occurs. Now let's discuss postural and phasic muscles. Each major muscle is designated as postural or phasic. Postural muscles are essential in maintaining upright positions, while phasic muscles are primarily involved in movement. The proportions of type 1 and type 2 muscle fibers may have slight differences in terms of cellular composition. So the postural muscles tend to be high endurance type 1 fibers. They adapt to stress by tightening and weakening, while the phasic muscles are fast twitch, low endurance type 2 fibers, and they adapt to stress by shortening and tightening. The postural muscles are the upper trapezius, the levator scapula, the latissimus dorsi, the erector spinae, the sacrospinalis, the quadratus lumborum, the piriformis, the sternocleidomastoid, the upper pectoralis major, the pectoralis minor, the iliopsoas, the medial hamstrings, the adductor longus, the oblique abdominals, the tensa fascia lata, the rectus femoris, the soleus, and the gastrocnemius, the tibialis, anterior and posterior. The phasic muscles are the scalenes, the deltoid, the biceps and triceps brachii, the lower pectoralis major, the middle and lower trapezius, the rhomboids, the serratus anterior, the rectus abdominis, the gluteals, the hamstrings, the vastus muscles, the perineals, and the elbow extensors. Okay, so we've reached the end of today's lecture, and for assignment number 8, I'd like you to list and give a brief explanation of the 3 mechanical properties of fascia, briefly define and explain the difference between motor unit recruitment and muscle recruitment, list the distinguishing characteristics of a trigger point, outline how to use reciprocal inhibition to relieve a cramp in the hamstrings. And as always, if you have any difficulty with this assignment, you may email me or text me. And of course, my office hours are on Thursdays from 4 to 6 p.m. If you have any difficulty at all, please do not hesitate to contact me. And with that, I will see you in the next lecture.